CYT is your term for cell. So if you see anything after CYT, it's referred to as cell. Cells are referred to those smallest things that are still considered living. And they're full of all these different things called organelles. So the thing about cells is they all pretty much have the same exact organelles, but they appear, they have small differences. So a neuron looks different than a muscle cell because it is differentiated. I mean, it's developed some small differences, but it maintains the same sort of inner machinery. So cells vary in size and shape, structure and function are interrelated. What the heck does that mean? That means when you see interrelated, so the structure of this kind of tells you about what its function is. It's one huge just storage circle. Okay, so it's going to store a bunch of carbohydrates and triglycerides. What kind of cell would this be?
Fat cell.
A fat cell because it's big, it's round, and it holds stuff. Okay, this cell has extended portions.
Nerve cell.
That's a nerve cell, and so just think about why it's shaped like that. Well, if you want to get power to your house, you have to send out a power line, right? So you have to have this long process from the power plant to go to your house. It's the same thing with neurons in the body. That's why they're shaped like this. But if you look, you see a nucleus, you see a cytoplasm, and you see a cell membrane in both of these cells. Okay, same thing with this is a muscle cell, this is another type of muscle cell, and these are squamous cells. So we're going to be talking about shapes of cells tomorrow in lab. And the flat ones are always the squished ones are the squamous cells. That's how I remember squished and squamous. We'll start with SQ. U. Cool. Three parts: nucleus, cytoplasm, cell membrane, as we just mentioned. So those, you have to have all three of those to be a cell. Okay, if you don't have one, I take that back. Okay, there's one cell in the body that does not have a nucleus, but is still considered a cell. Does anybody know which one that is? This is like a Jeopardy question.
The ovaries?
Not the ovaries. The oocytes are actually humongous, like the largest cell in the body, and they do still have a nucleus. A good guess though.
Brain.
Brain cells still have a nucleus. It's a good guess though. What cells last 120 days? Blood cells. Okay, so remember that blood cells actually have a lifespan. They are not immortal. They last only 120 days because they don't have a nucleus. They don't have the control center. And that is the only cell without a nucleus. Most cells in the body do have this nucleus, which is the central shaped portion that contains this stringy macromolecule. What's maintained within the nucleus? DNA, good. Ribosome parts, RNA, some other stuff. You see all these other organelles suspended within the cytoplasm, which is the fluid inside the cell. So these organelles all have names, they have a specific shape and generally one specific function. So it tends to work out pretty well when you're studying because you can just make one note card for every organelle. On the front, put the name; on the back, put a picture of it. Like these, to me, they look like circus peanuts. As soon as you hear that, you're like, oh yeah. They're like just regular peanuts, whatever. You know, these are the mitochondria. They look like peanuts, and I guess peanuts kind of give me energy when I eat them. Mitochondria are there to give you energy, you know, to supply you with energy, so that might work. And you can come up with generally like a silly analogy for each one. The centrioles, they look like little, you know those little Twizzler parts, like the Twizzler bites?
Mm-hmm.
That's what the centrioles look like. And we got these different cell parts. So rather than spend a bunch of time on telling you about each organelle, I'm just going to kind of give you guys some, since each organelle has one major function that's generally pretty basic, we're just going to do an activity with the organelles instead of looking at the slides. So when we get to that, we'll get to it. The outer boundary of the cell is the plasma membrane. which is a word I really struggle pronouncing still. You know how like you've got those words that you just can't say, like the couple words in your vocabulary that just don't come out right? For some reason, for me, I can't put plasma and membrane together. Try it real quick. Plasma membrane. That sounds kind of slow. It's okay. Yeah, so you guys can laugh at me for that. Okay, anyway, integrity of the cell is maintained by that. Remember that plasma membrane is built from phospholipids, and it allows some stuff in and some stuff out. It is selectively permeable. So that's the term you're going to hear a lot when you talk about let's go with cell membrane. When you talk about the cell membrane, you're going to hear it's selectively permeable. That just means that it has the ability to control what it takes in and what it spits out. And it does that using protein channels. So we'll see lots of stuff in that membrane. Protein channels, lipids, some carbs. And another bold term you're going to see associated with the cell membrane is signal transduction. That means it has the ability to send and receive messages. So the cell membrane has like mailboxes, sorts of structures that have the ability to receive messages and also send them out. And we call those receptors whenever we talk about the cell. So we have to take a look at the details of the bilayer. You know from last chapter that it's formed from fat cells. So those are called phospholipids. One part, it is actually a bilayer of those phospholipids. So we get 2 layers of phospholipids. In terms of the structure of the phospholipid, the head is going to be water soluble or hydrophilic, and the tail is going to be water insoluble or also known as hydrophobic. So you know what parts are going to point towards the water and what parts are going to point away from the water. There's a bunch of stuff in there, like cholesterol helps to stabilize the membrane. We talked about cholesterol as a ingredient to build hormones, but now we see another function is to stabilize the membrane. It contains membrane proteins, which have many varied functions. So we're going to talk about proteins in a little bit of detail today. Membrane proteins. Each one serves a purpose. We talked about how proteins were built last week. So just basics. What do you have to start out with to get one of these proteins? What are the building blocks of proteins? Amino acids, right? And we start to link them together in different like bonding patterns. Remember, the chain is just the primary structure. As soon as we get the helices and the sheets, we know we have that secondary structure. So all proteins are built in the same manner, but they all have a different final form. Right? And some of them can act as enzymes. Some of them are receptors. Most importantly, I would say the stuff that we talk about the most are protein channels. They're like little tunnels through the membrane. Each channel is a tunnel for a different substance. You've got channels just for chloride. You've got channels just for getting potassium into the cell. You have channels just for shuttling potassium out of the cell. And so as we go along in anatomy, we focus on individual channels and kind of what their purpose is or what their role is. So just as an example, this little purple thing is a protein channel. Anything purple, labeled purple, embedded in the membrane is going to be built with protein. This one, if this were a chloride channel, which means you've got a channel that just sends chloride through the cell. Remember when I think we talked about cystic fibrosis. We may have mentioned it, but that's a disease where your chloride channel is not built properly. So you're born with this mutation where your chloride channel just doesn't get built right because your DNA is a little bit messed up. And now you kind of have a channel that doesn't easily open and close. It's kind of busted and broken, okay? So now that you have a broken channel, I work with what I got, guys. Now chloride cannot freely move into and out of the cell, and it causes all those issues that you know as cystic fibrosis. Just one channel mutation causes mucus buildup in the lungs, digestive issues. You know, the lifespan is like, your average lifespan for cystic fibrosis is like 30, 35, just because of one channel, which is really unfortunate, but it tells you how important those protein channels are. And carbohydrates are also important to the cell structure because they help with, let's see, so conditions like lupus. Do you guys know what happens to people with lupus? What's going on in somebody's body when they have lupus? Somebody raise their hand and tell us about that. Sabrina, go ahead.
It's autoimmune.
Autoimmune, yeah.
Your body basically starts attacking itself. It starts attacking hair growth, so people lose their hair. It starts attacking joints sometimes, so they might develop like more severe arthritis. But as in terms of like what particular cell it attacks, I'm not sure.
Yeah.
But I just know the immune system is overreacting, so the person has heightened levels of white blood cells. But in terms of which particular blood cell, which particular white blood cell, I don't know.
Yeah, so the body attacks itself, and it attacks almost every organ. If you've got the bad kind of lupus, all of your organs it's like a mutiny. But it's an inappropriate mutiny. Exactly what's happening is these cell recognition structures or self-markers embedded in all the cells in the body, they are called glycoproteins. They act as your cell's ID tags. All your white blood cells have a picture of what your cells look like bouncers at a club. They check the ID. If it's a fake ID, you're gone, right? You're kicked out. Unfortunately, in somebody with autoimmune, their white blood cells mistakenly think that those are fake IDs. So they can't read your own cell recognition markers. So they start attacking your cells because they think they're foreign, right? So that would just be like if you went to the bar and you had your real ID and the bouncer's like, no, this is fake. You're adding it. Like, no, but this is my real ID. So that's how all the cells in your body are feeling, because they are the real ID tags. It's a weird thing. Nobody understands autoimmune. Autoimmune is the weirdest disease, and maybe you haven't thought about this before, but go to like a third world country. People don't suffer from autoimmune. Their bodies are too busy fighting off real diseases. whereas like malaria and stuff. Whereas in the U.S. Or a developed country, our bodies, I think they just kind of get bored and they start to act serious. I think they don't have anything to. The immune system doesn't have anything to do, so it just starts attacking their own cells. If you look at autoimmune in the U.S. versus just any developing country, it's going to be just a stark difference in the rates.
Is it something, are you born with it, or is it something your body has to develop?
It can be either. You can be born with autoimmune, like type 1 diabetes is a mutation in your genes that codes for damage or destruction of the pancreas. MS is autoimmune, which you develop later in life, right? Yeah. Nobody understands autoimmune. It's really kind of a mystery. Something to think about. Okay. Cytoplasm, fluid, there's stuff in it. We call those organelles. They're little tiny structures with specific jobs. So we're going to talk about every organelle in detail in our little activity. The cytoplasm contains the cytoskeleton, which is a support framework of protein rods. So if you look at a building being built, you see all the truss rods and 2x4s and all that stuff that makes up the solid framework of the building. That would be the cytoskeleton in the cell. And now we're going to go through the organelles. So for these next, I think, 10 slides or so, I just want to talk through the organelles. Is it the organelles, believe it or not. Okay. So the rubber chicken. Chickens lay eggs all the time, right? There are structures in your body called ribosomes that lay down protein. Okay. So get the rubber chicken. You need people on opposite sides here. You don't want to know what this is from. I didn't use it. I used these. Okay, and then the batteries are. What do you think the batteries are, guys? Mitochondria, they're good. Okay. So you're going to see rough ER and smooth ER. A recipe. Rough ER and smooth ER as well. This is going to rough ER and smooth ER. Okay. And then the lysosome. What are these for? I don't even remember.
Are your eggs from your chicken?
I guess I'll get to them later. Oh, you're right. The chicken has to lay eggs. Okay, and then Lysol is, you might be able to guess what that is. Okay. So, as I said, I'm gonna leave the making of the note cards to you guys. I just want to take you guys briefly through the function of each organelle and then do a little activity to try to help you get a visual of what's happening in the cell. So the cookbook is important. Hannah, I can do that. So if you flip through all these, each slide is on a different organelle. I just want to go back to the beginning. Here, so this is the big picture. The nucleus is in the center of the cell. The nucleus contains the DNA. The function of the DNA is two major functions. So we have protein synthesis, which is the creation of proteins, and then also passing on the genetic information. Most of what that DNA is doing at any given time is the former, is protein synthesis. So passing on instructions to build proteins. And those instructions are maintained within the DNA, the code of the DNA, which is why I think about the DNA as a cookbook. I don't know how many people in here like to cook. I think cooking is a fascinating science. I love cooking. I think it's a fantastic thing. I think it opens up your brain to all these different tastes and your smells, and you're practicing following directions, which I have trouble with in general. So cooking is really neat. And in the body, our cells are cooking all the time. So the DNA contains all the instructions to make proteins. So Hannah, open up that cookbook just to any page, and we're going to talk about how a protein is built using the function of the organs. Whatever recipe looks good to you.
Marinated chuck roast.
Okay. Yeah. Maybe that's what this is for. I still don't know what that's for. Marinated chuck roast, great. So just picture that is the chloride channel. The chloride channel needs to be built. Okay, chloride channel is protein based, right? So the instructions come from the DNA. This is how you make marinated chuck roast, right? And then what Hannah does is she writes down the recipe on a note card. The note card is the sequence of amino acids required to make that chuck roast. Okay, and for our purposes, we're going to pretend that's that protein channel that we need to shuttle in chloride. Okay, so if you look at the way the cell is set up, it's actually really intuitive. It makes sense. So it may look like a jumble of stuff initially, but this right here, directly surrounding the DNA, is the rough endoplasmic reticulum, and it is the organelle that receives the instructions to make that protein. And it receives those instructions because it's right next to the nucleus. So we have just a small little path of travel for those instructions to get there. So go ahead and pass those along to the ribosome. Actually, all you gotta do is pass the recipe. Because think about this also, the DNA has to stay in the nucleus. So it needs a messenger to get the DNA, to get that code to the ribosome. That's called RNA. When we talked about RNA, we said, hey, this is like DNA, but it's single-stranded. This is what we're using RNA for. It's acting as the messenger. So RNA is sent to the ribosome. You're going to read, which is the rubber chicken. It's the rubber chicken because rubber chickens are awesome, and because rubber chickens lay eggs, right? So you read the recipe, and then rubber chicken, go ahead and hold up your rubber chicken so everybody knows. Okay. That rubber chicken is representing the ribosome. Ribosome will lay an egg. Okay. So we laid an egg. That egg is picked up by the next organelle, which is the rough endoplasmic reticulum. So rough endoplasmic reticulum is, see how it looks like a series of canals here? Technically, the ribosomes are embedded on the walls of that organelle. So as they start to take in the instructions and spit out protein, that protein is picked up by the rough ER, which is this series of canals here. Okay, so that amino acid chain that was created by the ribosome is then picked up by the rough endoplasmic reticulum. So you're going to have to fry this egg just a little bit, okay? Just pretend like you're frying an egg. Just frying that egg, that's representing the rough endoplasmic reticulum making small chemical alterations to the protein. So she starts to fry the protein or fry the egg. She passes it on to the plate. So, yep, just the egg. Awesome. Okay. We skipped a step here. The analogy's not perfect. Next, what's supposed to happen is the protein is going to go through the smoothie yard. Its destination is the membrane, cell membrane. So it goes from the DNA to the ribosome, through the rough ER, frying it up. Should go through the smoothie yard next, okay, but we skipped that. We went straight to the Golgi. The Golgi is a specialized apparatus. Does anybody know what the Golgi's purpose is? How much of Learn Smart do you remember? Yeah, go ahead, Oscar.
The Golgi pretty much transfers the proteins. It's like how things get in and out through the membrane. Some proteins.
Yes, yes. So the key word was transfer. The Golgi is like a packaging and shipping plant for proteins. Okay, so when I think about the Golgi in terms of our food analogy or our cooking analogy, I think about the Golgi as the plate. So in order to get that entree out to the table, you need a plate. Okay, so to me, go ahead and hold up the plate with the protein on it. That to me is the Golgi because that's what picks up and ships the protein out to where it needs to go. So we have a series of steps here before that protein can be delivered to the plasma membrane that's going to perform that function. So, you know, we can pass around that protein and eventually we can get it here to the plasma membrane. You have to use your imagination a little bit to picture this turning into this. But that's the final goal, right? Is to get fully functional built protein channel, like the chloride channel. And that's the function, the major function of each organelle along the way is covered. So let's do it one more time. And I'm going to let you guys who have the organelles, will help you out if you don't know, tell me what's going on. So first step, Hannah, you are the
nucleus, so for the DNA, and then the recipe would be the instructions. So I'm sending my instructions to her, which is the chicken.
And those instructions, to clarify, what molecule is that? It's not the DNA, it is the RNA. So that's the messenger. That is the recipe that's been
only
copied.
carry one chromosome from the 26, right? Or from the 40.
The RNA has the ability to copy any of the chromosomes it wants to. But the RNA specifically is just a single side of the DNA's code. Yeah, that's what you're going for there. Okay, and then what's next? You take those instructions, you read them.
Yeah, and then it shoots out your, I don't know what you call it, like the egg, which is.
Protein, yeah.
Protein.
Yep, so you lay the egg, that's the protein, and then, yeah, and which ER are you? Rough ER, good. So fry the egg. You're doing some, and it's not easy to fry an egg properly.
Well, that's
right?
what wants to stand up.
Yeah, I know.
So-
Yeah. Like I said, we work with it.
So then
Now what do you do?
it goes to the
So it would have gone through the smooth ER
Golgi.
and then to the Golgi. We're going straight to the Golgi. Well done.
And then it's like taking out food at a restaurant and I'm transporting it to the plasma membrane.
Good. So the Golgi is that you're kind of like, you're almost kind of like a chef though, because you take it and you refine it and you like garnish it or whatever you wanted to do to the egg. I don't know if anybody garnishes eggs. Maybe you're making like a fancy, eggs Benedict or something like that. But you garnish it, you put all the trappings on it, and then you package it, and then you send it out right, to where it needs to be. So we have these, which of course this becomes the fully functional protein chain magically. We have these structures in the body that actually do the shipping. They're called vesicles. So when you hear the word a vesicle is a name given to any just bubbly sac, membranous sac inside the cell that ships something around. It's like the Uber, it's like a taxi or an Uber for anything in the cell. So if you hear the word vesicle, you just want to think like an Uber or a shipping vehicle in the cell. They all just have this boring circular membranous shape.
Goal purposes to just transfer.
Yeah, anything. It could be anything. We've got a special one, and they all have, a couple of them have different names, but we named this one specifically, this vesicle here. It's a little bubble that transfers stuff around the cell. This is called the lysosome. So Lysol, you've got our Lysol right there, right? If something bad entered the cell, like a bacteria or a virus or anything that doesn't need to be there, even just debris or like dirt. I don't know. Dirt doesn't really go into cells, but like cellular dirt. Anyway, anything goes into the cell that we don't need. What the lysosome does is it envelops whatever that is, okay, like an amoeba sort of, and then it's got these enzymes, which, to me Lysol is a perfect name, because whenever you want to degrade or get rid of bacteria or viruses, you just kind of like wipe the surface of the table with Lysol. Whenever you want to degrade or take something out in the cell that shouldn't be there, you use the lysosome. It does the same thing.
The janitor.
Yeah, it's like the janitor. It wipes everything down so we don't get Zika. Nobody's gonna get Zika or Ebola. I was talking to one of my buddies. His wife is a researcher. She does like disease research for like the CDC and whatnot. And I was like, okay, so let's just hit the major topics here. Is anybody gonna get Zika? And she's like, no, nobody's gonna get Zika in the U.S. Is anybody gonna get Ebola? She's like, no. And I said, how many confirmed cases of each other? And she said, you couldn't even count it on 10 fingers. And how many, billions of people are, populations in the billions, right?
7.4.
Not a geographical specialist. 7.4 billion people, right? What are your chances of getting Zika and Ebola? Absolutely nothing. So just a funny conversation. But that's the general gist of what the organelles do. If you want to get a deeper picture, make sure, as you always do, or as you're doing all the time, is reading the textbook, okay, in detail. That tells you everything about them. But like I said, in general, one organelle has one specific function. So I want to lastly talk about the fishing lines.
I have a question really quick.
Sure, go ahead.
So whenever that RNA carries those instructions, does it always skip the smooth
Mhm.
plasmic reticulum?
Nope. What happens is sometimes it just depends on the protein that's being built.
Okay.
The more complex proteins must be sent through the smoothie endoplasmic reticulum, but some don't have to go through there.
Okay.
Some go straight from rough yard to Golgi, frying pan to plate.
Okay.
Some go from a frying pan to another frying pan to a plate. Right, more, like a more complex recipe, you wouldn't just be able to fry up and put on a plate. You might be able to do like an egg, but that would indicate kind of the function of the smoothie R is to make another change or make another alteration
Okay.
to that growing protein. So you don't always use it.
Okay.
Any other questions about the basics of organelles? Okay, I want to talk about mitosis for just a bit. There's, like I said, we've got these organelles that look like Twizzler bites, and they are together. They're called the centrosome. They consist of two centrioles. So Oscar and, I'm sorry, what's your name back there in the corner? Jamie have these two fishing poles. These are centrioles individually. Centrioles, you put them together, they are the centrosome. The centrosome, or the centrioles, only function during mitosis. What they do is they throw out these long fishing lines called microtubules, and they reel in the DNA to either side of the cell. When would you reel in the DNA? You'd reel it in if you wanted to duplicate the cell. If you wanted to take one cell and make two, you have to take the DNA in that cell and duplicate it, so you end up with double that genetic information. Once you have two copies of, and we're going to talk about this in more detail today, once you have two copies of the DNA, you have to have a way to get them to either side of the cell so we can split that cell in half. And that's the purpose of the centrioles. So the reason I like to think of them as fishing poles is because they throw out, fishing pole reels in the fish. The fishing line indicates, so go ahead and actually give us a visual. Pull out that fishing line a little bit. That line right there is indicating, the microtubule. A microtubule is just one long protein that can latch onto stuff and move it around in the cell. So really I want you to just visualize that process as long fishing lines reeling in the chromosomes. And I'm going to try to show you guys some YouTube videos and animations as well. So that we just discussed is a separate function of DNA. The first function that we talked about with making the protein, that was the first major function of DNA, protein synthesis. The second function of DNA is replication, or mitosis. And that's when you centrioles come into play. You don't have any, we're not using the fishing poles whenever we're cooking. Think of any recipe that would involve fishing line. But whenever we're performing mitosis, we need those centrioles. Okay, questions so far? How much did I'm curious because I didn't do this assignment. How much did the Learn Smart go into detail on organelles? Did it go through pretty much every one and just kind of touch the basics?
Like everything we just talked about.
Yeah.
Pretty much just went through the basics.
Pretty much the basics of the organelles. Okay. Yeah. So the reason I do this is so you can take that information and put it together in one thing, right? The big picture. Don't miss what do they say? Don't miss the forest for the trees, right? So don't leave out the big picture for all the details. And that happens all the time in anatomy because you've got all these details and you have to somehow put them together. So this is kind of how I like to do that. So I hope that helped a little bit. The cilia, these are some things that we didn't talk about, the rest of the slides. So flip to cilia. The cilia are the hairs that hang off of some cells. You only have them in a few places in the body. The major place that we talk about cilia is the respiratory system. So inside your airways, right, your trachea will split into two major tubes that go into your lungs. We call those the bronchi. When you look, if you were traveling magic school bus style,
Yes.
if you were traveling into the lung through one of those airways, I know you guys were thinking that. And you know, you were bus, and you were just like, you were driving down the airway road. You would be driving through, if you're driving on top of those cells in the airway, you would be driving through like reeds, sort of. So those would be the cilia, and these extensions that hang off the cells, and they have the ability to sweep things around. So all the cilia in the respiratory system are constantly moving, and they're sweeping stuff this way, sweeping stuff out. So the function of the cilia is to propel mucus in the respiratory tract. And they beat back and forth in a very coordinated manner. So all of those cilia at once and all of your airways would be beating this way and sending mucus out. Why?
All day long.
Why do we do that? Go ahead, Joe.
Oh, no, I was just saying they catch crap all day long.
Yeah, that's all they do. They catch crap that's in your mucus. I mean, think about all the stuff, all the crap that we inhale, right? I mean, hopefully not physical crap, but like, you know, talking about like debris and irritants from the environment. Talk about all the stuff that's in the air. Right? There was just, as you guys know, I live in Indy. We weren't supposed to go outside yesterday because
Air
there was
quality.
so much crap in the air.
Yeah.
Yeah, air quality day or whatever. So, you know, that tells you how essential cilia are. If you've got all that stuff going down, you need a way to get it back up and out. They can become extra active when you have sickness, because now the cilia know that there's some sort of bacteria or organism inside the mucus that lines your respiratory system, so we need to be even harder to get that virus out. Talking about smoking, this is, I think, my only smoking plug. I don't really talk about smoking a lot. Everybody has their habits. And I've got one bit of information on smoking. Smoking actually, over time, it will paralyze and damage the cilia. So it's not like, the smoking doesn't, think about how essential the cilia are to keeping your airways healthy, right? If the cilia don't work, then your airways are then going to experience severe irritation all the time. So that's what smoking does, is it paralyzes the cilia. Quit smoking, the cilia to a certain point can regenerate, but they'll never be the same if you were a smoker for like 30 years. Right? So keep that in mind.
Is that why they develop the smoker's cough? Is because they're trying to,
Yeah.
they're not activating, so they have to cough really hard to get all the crap
Exactly.
out.
Yeah, so now the cilia don't do what they should. Now you've got to really like hack up. You got to cough really hard to get that stuff out because the cilia aren't there to do it anymore. That's just one of the things that comes along with smoking. Yeah. So I don't get on a soapbox or anything, but I do like to educate people about, you know, what's going on with smoking. So one other area is the uterus. If you think about the fallopian tubes, whenever fertilization occurs in the top of the fallopian tube, it doesn't happen in the uterus. The egg has to travel a really long distance to get from the top of the fallopian tubes into the uterus to implant, and it only gets there if the cilia in the fallopian tubes sweeps it down. And that process still takes 10 days sometimes. Did you know that? So 10 days after fertilization, the egg implants in the uterine wall. That's on average. Yeah, you just wouldn't think it would take that long, right? Because it's just this little distance, but it's a long way if you're just a little egg cell. So cilia are also there, and I think those are the only two examples that we'll give. Cilia, that is the medium-sized cell extension. The longest one is the flagella. The only cell in the body that has a flagellum is a sperm cell, because it has to be able to swim a pretty long distance. I think the relative distance it has to travel. We just said that fertilization happens all the way up in the fallopian tubes. The sperm cell, that's like a marathon for sperm cell. That's like 26 miles relative. So it's going to need, you're going to notice it's got this long tail, which is the flagellum. And then it has a midpiece here. The reason you see that kind of bulbous area is because you have a midpiece full of mitochondria, which you now know the batteries, right? Hold up your battery, people who I gave batteries to. You guys are powering the cell. The battery is making a molecule called go ahead, Joe.
ATP.
ATP powers every cellular process. Think about how big that flagellum is. It's going to need a lot of power. It's like a propeller. A lot of sperm cells, a lot of bacteria cells have flagellum. Okay, so that would be, in terms of outside the human cells. That's who else has them. Okay, fibers. There are three main types of fibers that hold your cell together. So we're talking about the cytoskeleton here. The cytoskeleton is the framework that holds your cell together. Microfilaments are tiny rods of actin. They're both of actin. They provide cellular movement. So we're really going to hold off on talking about microfilaments until we get to the muscular system. Your mantra in the muscular system will be actin and myosin, actin and myosin, actin and myosin. What the heck are those? They're called microfilaments. And in the muscular system, they are the ones that provide that movement of those muscle cells. Microtubules are a little bit larger. They are built from, as the name suggests, tubulin, parts of tubulin. Tubulin is actually a structure or a molecule that makes up cilia, the flagellum, and the centrioles. So if you cut up a cilia or cut up a flagellum, you looked at it, it would be built of tubulin. So the microtubules are the largest, those largest filaments inside the cell, and they latch onto and also move organelles around. So if this mitochondria needs to move to make way for another mitochondria, say we're making more mitochondria in the cell, then this microtubule will actually pull that mitochondria over this way into place. So not only do they hold them together, they also have the ability to shift them around. Okay, it's like the roof in Lucas Oil Stadium. It's a movable roof. I don't know if anybody's been in there. I'm sure some of you guys have. This is Colts land, right? It's still Bloomington. The roof moves, right? So it will open and close, but it also holds the solid structure of the building up. And intermediate filaments are composed of several proteins. They are most specifically associated with supporting the nuclear envelope. So the nuclear envelope is right here. That's what maintains the shape of the nucleus. So if you see a filament running into the nuclear envelope, it's generally going to be an intermediate filament. This is just the nucleus. Don't get confused. This is not the whole cell. This is just that center part. The thing that makes the nucleus special is it's got a quadruple layer of phospholipids. So I want you to distinguish between this, which is 4 layers of phospholipids, and just this, which would be 2 layers. That's what the cell membrane. If you're looking at the cell membrane, you would only see this. If we're looking at the nuclear envelope, so you'd see 4. Chromatin is inside the nucleus. Chromatin is just a fancy name for jumbled DNA, or disorganized DNA. So if you hear the word chromatin, we're still talking about DNA, we're just saying this is disorganized form. So as we go into a little bit more detail on mitosis, we're going to mention that chromatin, right before a cell begins to replicate or duplicate itself, that disorganized chromatin that looks just like a bunch of strands, like spaghetti right now, will organize itself into 23 official pairs of chromosomes. And that is the organized form of DNA is chromosomes. So if you want to write yourself a note, just to help you remember, if you wrote chromatin as the disorganized form, you might want to write chromosome as the organized form of DNA. That might already be on here somewhere. So those are the basics of all the parts of the cell, guys, so far. We're going to talk about a little bit more of a challenging concept, and that is transport. the reason this becomes challenging is because a lot of these processes have similarities, small similarities and small differences, and you have to remember all the similarities and differences, so it gets a little bit tricky. So the first thing we do is just kind of group these. Passive processes, or transport, we're talking about moving stuff into and out of the cell. Passive transport does not require ATP. So mitochondria, hold up your batteries. When we are performing passive transport processes, mitochondria are not needed. It's like a toy that doesn't need batteries to work, okay? That would be a passive process. You can back down now. Your active processes are transport processes that do require ATP. They would be like the fancy toys, right? That require 90 batteries to work. So you've got some processes in the body that require more energy than others. Active transport does require the functioning of mitochondria. And these are all the active transport processes. Do yourself a favor and remember when I told you guys about mnemonics or memory tricks? Make a memory trick or make a mnemonic for the passive processes and the active processes. So take those letters and arrange them in an easier format. Or make a sentence using the first letter of all those words. I want you to try to do that really quickly with your neighbor. Just take a minute or two or so and come up with a way together to remember these letters in order, or a sentence. And then I want to ask somebody to share because I don't have a good one for this, so I gotta get one. Maybe something that has to do with like like eating, so like as an active process. So you put those two words together, you might be able to do something like that. Talk about it just for a little bit, see if you guys can think of something. Anybody? I think I'm just gonna go with, so for the active ones, the way you know it's active. Just add another E onto the beginning. Okay, that'll help me. Hopefully that helps you. And then passive. Anybody have a way for that, to remember that one? No, it's kind of weird, yeah.
I was thinking, did you think of it going down, like not going against, but going with the flow?
Oh, okay, so you're talking about the process itself. I'm just talking about a way to remember the letters involved.
I can't think of anything,
Okay.
but let's think of go with the flow.
Okay, gotcha. Yeah, go ahead, Joe.
For active, like I kind of switched them around and I put like ETA, as in like an arrival to an airport.
Oh, ETA, yeah.
Yeah,
estimated
so
time
like,
arrival.
yeah, because it's active and you think of flying as being
Okay.
kind of active.
Yeah, I like that.
Only I put the other E at the beginning, so it's like ETA.
Expected time of arrival.
Expected time of arrival.
Gotcha. Cool. Yeah, so that might, if you can remember one, a lot of times you can remember the other one. So hopefully just getting that one down will help. We're going to talk about each one of these processes in a little bit of detail. So just keep in mind the first four do not require ATP. And the first one is diffusion. So diffusion is specifically just the regular spreading out of matter. just in the general sense of, in the most basic sense of chemistry, it's just a spreading out of matter equally. If you want an example, it would just be, you know, sugar dissolving or food coloring dissolving into a liquid, any molecule spreading out evenly. I think in physics, we call this entropy. If anybody's familiar with physics, I'm not, so I was never a physics person, but I do know that entropy is this spreading out of matter. So this occurs due to constant motion of atoms, molecules, and ions. The speak that we give it in anatomy is moving from a region of high concentration to a region of low concentration. So whenever you're talking about a cell, and I like to put everything in terms of a cell because we're in anatomy class. If there is a substance out here, okay, little molecules, and there are a bunch of them out here, and there are hardly any in here. This is a high concentration of that stuff. And when we talk about stuff, we're talking about a solute. Just think stuff. There's a high concentration of solute out here. Brackets mean concentration. There's a low concentration of solute in here. Once again, brackets mean concentration. So naturally, if the membrane is permeable to this stuff, it will balance itself out, right? Homeostasis, a balance, an equilibrium in the body. This is just the homeostasis of chemicals. It's called diffusion. The only stuff that the cell membrane is freely permeable to, meaning it will move freely without any help, is oxygen, carbon dioxide, and other lipids. So why is this? Why are these things free to move through the cell membrane without, they get a free pass? Any ideas? What do those things have in common?
They are all fat soluble, so the phospholipids will allow them to come in and out more freely than your sugars and other molecules that are not fat soluble.
Right, so the cell membrane is made of fats, phospholipids. These are all fat soluble, meaning they dissolve in fat. So, since the cell membrane is fat and these molecules are fat soluble, they have the ability to pass freely. So that's kind of your rule, is if something is fat soluble, then it will pass through, or will permeate freely through into and out of the cell. So you just want to add these to your list. Stuff that can pass freely. Dissolving sugar cube is an example of diffusion as well. And I'm thankful that these are fat soluble, oxygen, these molecules, because if they weren't, then your cells would not receive a constant supply of oxygen if oxygen was not fat soluble. So it would take energy and work to put a protein channel in the membrane and pull it in and deliberately do all this stuff just to get oxygen in and CO2 out. Fortunately, those molecules move freely, so it doesn't take extra work for that diffusion. In terms of like what's happening in a beaker, something you could do in a lab, we gave the example of a cell. I like this example better because it's an application. But these beakers are representing two columns of fluid separated by a cell membrane. That's what this little membrane is representing, is the cell membrane. Basically what's happening here is it's showing two types of molecules, a solute molecule, which is the bigger one, and then a water molecule, which is the smaller one. And we've got, if we go from right to left here, the membrane is permeable to both water and the solute. So there's a high concentration of water over here. Remember we said diffusion is a substance going from a high concentration to a low concentration. If this membrane allows both water and the solute through, we start out with more water on this side than this side. So if we travel, we allow time to pass, the water equalizes because it passed through the membrane, through diffusion. And then I want you now to just look at the orange circles. Same thing happens with those. As time passes, the orange circles will also equalize on either side of that membrane. So equilibrium is reached when the concentrations are the same. That's how the body works. This is why if you drink enough water, you'll die. Let's talk about that. It's called water intoxication. People who go to like what are those things called? Electric Daisy Carnival and the stuff that, you know, like the big raves, like the scheduled raves where you're supposed to bring ecstasy and stuff, right? That stuff, so whenever people consume like high amounts of ecstasy and Molly and MDMA and that stuff, what they start to do is their brain starts to crave water because they get really dehydrated, but then they overcompensate and they drink so much water to the point they die. It's called water intoxication. It's based on diffusion. So when you put that much water into your body, you have trillions of cells in the body. Yeah, I'll get to it in a second, Alex. You put that much water in the body, you're loading the outside of your cells with a ton of water. What's that water going to want to do?
Cross the membrane.
Cross the membrane and balance out through diffusion. What happens when a cell reaches a certain swelling point? It bursts. So what the reason you die is because your brain cells burst. That sucks. Right? That's why you shouldn't do ecstasy.
That's the reason.
Like I'm in dare program. Yeah. Love it. Yeah, Alex, is your
That was
question?
my thing. That was my thing. The
Yeah.
fact that you put something in so much water that
Yeah.
the water's gonna want to reach equilibrium, and then
Yeah.
it will just cause the cell to burst.
So that also helps us understand like how how IVs work. Like when you guys actually go into nursing and you start to study IVs, this information is so important because you have to understand concentrations of certain liquids you put in the bloodstream. If you put in a liquid that is a high water content, right, then what you're going to do is you're going to hydrate the cells. Sometimes you don't want to hydrate the cells. You want to put in a liquid that's at a lower water concentration. So this gets so much more complex, but these are the basics. Think about diffusion, and what will that substance do to equalize itself out? Okay. Sabrina, yeah?
Would it be appropriate to use the term hypotonic and hypertonic?
It would, and we will in a few slides. So that way we can actually talk about, okay, so what is a hypotonic solution and what is it going to do to the body and so on and so forth, which is stuff that you need to know to pass the NCLEX. And a cell is placed in a hypotonic solution. How will the cell respond? So yeah, talk about that too. And that was all diffusion. The reason we talk about diffusion for a while is because it's important in terms of IVs and whatnot. Facilitated diffusion is the same concept. These molecules are at a high concentration first outside the cell, than they are inside the cell. Those molecules are representing glucose, the red ones. And since glucose is water soluble and not fat soluble, it's not in that list, it's a special list. This is an important list. Since none of these substances, sodium, potassium chloride, glucose, amino acids, are fat soluble, they are going to need help crossing the membrane. So they need channels. In facilitated diffusion, the high concentration of stuff outside the cell to inside the cell drives the movement. So that means these molecules want to equalize. But there's a problem. they can't just cross through the membrane. They need a channel to cross. So when we put a channel in the membrane to allow them to cross, we call that facilitated diffusion. We facilitate the process. So think, if we have a channel there, it is going to be facilitated. Facilitated diffusion does not require ATP. You might want to write down another basic rule that most channels require ATP, but this is an exception to the rule. All the other times that we see channels, they are going to require ATP, so they'll be under the active transport. But these channels for facilitated diffusion don't require ATP. You'll get tripped up. Your brain will get confused if you don't remember that, because you're going to think channel ATP, but not every time. There's an exception. Sodium, potassium, chloride, glucose, amino acids. Osmosis is just the term for movement of water. We are specifically talking about H2O. So osmosis is the movement of water across a membrane from a higher concentration to a lower concentration. Water moves, always moves to the side that it wants to dilute. This is where things get weird, because now we're not talking about the solute. We're talking about, okay, where is water going to move? So it gets a little confusing, so we have to draw pictures. Osmosis is a passive process, and there is no ATP required for osmosis. Otherwise, we run out of ATP. Water, keep in mind, this is our general rule. Water always moves into a region containing more solutes, because water always wants to dilute the solutes. That's where this comes into play. This is a basic concept in nursing. This is probably your first nursing concept that we're going to talk about, and that is osmotic pressure. Osmosis is the movement of water. So osmotic pressure is defining water's ability to move. The pressure created by water's ability to move. So osmotic pressure increases as the concentration of solutes increases. Let's explain that with a picture. Okay, so there's water. I wish I had a blue marker, but I don't. Okay, so if a cell has a high solute concentration, remember that just means stuff. So we've got a bunch of stuff inside a cell, and let's just call that sodium. Any solute, any atom or molecule. And there's not as much stuff, the same molecule, outside the cell. Think about which direction water will move in this circumstance. Not the solute. We're just talking about which way water is going to move. Alex, go ahead. It would go into the cell. Your brain doesn't like that, because your brain's looking at these dots. Okay? Well, surely the dots are going to move out of the cell, because that's them moving from a high concentration. That's them equalizing. But we're talking about the movement of water. Water, in this circumstance, is going to attempt to dilute the high concentration of that stuff inside the cell. This circumstance, and it's going to cause that cell to swell. This circumstance is a hypotonic circumstance. So I want you guys to actually draw this picture out, because this doesn't help me, this helps me. Draw this out on your paper, and then we'll talk about it again. Yes?
Okay, so is it possible to say that water
No, water moves with or towards the solutes, right?
Yeah, but you're saying direction wise, so
Yeah,
you're
we-
correct in this circumstance because yeah, because as the solutes are moving this way, the water's trying to
Mhm.
dilute.
So
Yeah.
it's
It-
so diffusion and osmosis kind of happening at the same time.
They are happening at the same time. That's correct. But we don't talk about them happening at the same time because that confuses the issue when we're initially learning them. We learn this concept and then this concept, and then we learn, okay, this is all happening at the same time.
With that, with like osmosis, I think of like if someone's sodium level is high, you don't want to hydrate it because then they'll become overhydrated because the water will follow the salt.
Correct.
So, and
Correct.
then like when you sweat, you sweat out all the salt. So that's why you drink Gatorade and electrolytes and stuff because you're replenishing the salt that you just lost.
That's a great practical application of like things that we do naturally that exemplify this process. Yeah. That's what I always try to do as well. I try to put examples and for some reason this is just a challenging thing because your brain doesn't like this. It wants to talk about, okay, which way this is gonna move. But in terms of osmosis, we're always talking about which way water is moving in this circumstance. Water wants to go in and dilute the high amount of solutes inside the cell. Now, that's called hypotonic. Okay? That's a hypotonic solution. And this is the cell that's been placed in that hypotonic solution. So hypotonic is referring to the solution that we've placed this cell in. It's referring to this area. Okay. And next, let's do the opposite circumstance. You guys get the picture. Okay. So we have a situation where we have very few solutes inside the cell and more solutes outside the cell. Okay, so here, let's talk about our solute concentration first. We've got few solutes inside, lots of solutes outside the cell. And now let's focus on the spaces. So now let's look at water. So if we look